pardon me, we are going to start with a practice problem from the 2019 AP exam, and let's see if I can actually get this to show up the way that I want it to. There we are. That'll do. So this one is looking at action of an enzyme, it's looking at metabolism and energy, and it's looking a little bit at genetics. So in this one, we're looking at PDC, which catalyzes the conversion of pyruvate to acetyl-CoA, substrate for the Krebs cycle. The rate of pyruvate conversion is greatly reduced in individuals with PDC deficiency, a rare disorder. So for this one, we would need to identify where PDC is most active. So this goes back to dealing with glycolysis and the Krebs cycle. So all of this would happen in the mitochondria or the matrix of the mitochondria. So that's where that would be taking place. For the second part, that's asking us to make a claim about how PDC deficiency affects the amount of NADH produced by glycolysis and the amount of NADH produced by the Krebs cycle in the cell. We need to provide reasoning to support your claims based on the position of the PDC-catalyzed reaction in the sequence of cellular respiration's pathway. So, we know that at the end of glycolysis, we wind up with pyruvate, and pyruvate goes into the citric acid cycle. And going through the citric acid cycle, we get that reduced out to eventually carbon dioxide. It does some substrate level phosphorylation, gets us some ATP, and we also get NADH produced. It's an electron carrier and takes that to the electron transport chain. So we're going to look at this one. So PDC deficiency would definitely reduce the amount of NADH produced by the Krebs cycle because it is preventing us from getting the material that we need there. So the amount of NADH produced in the Krebs cycle would decrease because Krebs cycle uses acetyl coenzyme A, which is produced in greatly reduced quantities in cases of PDC deficiency. Let's try that again there. The other part of this one, asking about glycolysis. The amount of NADH produced during glycolysis would stay the same because glycolysis occurs before acetyl-CoA is before pyruvate is converted to acetyl-CoA. Part C says PDC deficiency is caused by mutations in the PDHA1 gene, which is located on the X chromosome. Male with PDC deficiency and a homozygous female with no family history of PDC deficiency have a male offspring. We need to calculate the probability that the male offspring will have PDC deficiency. So this is looking at an X-linked recessive trait. So in this case, what we're looking at, if a male with this trait has that, so his genotype would be X with a lowercase PDC gene, and he's got a Y chromosome for the other. And for the female, she is going to have 2 X chromosomes, each of those with functional copies of the PDC gene. So I'm using the uppercase PDC there to show that that is the functional copy. All right. So the possible male offspring for this, that male offspring would have to get the Y chromosome from the father. So he's going to have that, and he is going to get either X chromosome is going to have the functional copy of that. So there is going to be a 0% chance that the male offspring will inherit the disorder. So there we have that one. I will pause for a second so that we can get another one going here. All right, for our next one, it goes back to cell communication and the cell cycle. So this would be when we did that drug simulation that would tie in here. So we've got a neurotransmitter that is being released by a neuron and accepted by a synapse after there. And we have here acetylcholine is a neurotransmitter that can activate an action potential in a postsynaptic neuron. Researchers investigating the effect of a particular neurotoxin that causes the amount of acetylcholine released from presynaptic neurons to increase. So we need to, in part A, describe the immediate effect of the neurotoxin on the number of action potentials in a postsynaptic neuron, and predict whether the maximum membrane potential of the postsynaptic neuron will increase, decrease, or stay the same. So this is looking at some stuff that we haven't directly covered, which a lot of the problems that we're going to work with do. So they're going to be things that are related to what we've done before, but are going to require a little bit more insight and thinking. So as we look here, releasing the neurotransmitter causes, over a short period of time, the voltage on this membrane to spike and then go back down. So what we're going to be looking at here is what happens with this neurotoxin that increases the amount of acetylcholine on the action potential. So if this thing keeps getting stimulated and stimulated and stimulated, we would probably predict that those action potentials would come further or closer together. So for part A here, those action potentials will be closer together. This is because the postsynaptic neuron has an action potential exposed to acetylcholine. The other part of this, and this is where we didn't spend as much time looking at the mechanics of these action potentials. So if we notice, before it resets to normal, there's a little bit of a delay where it dips down below. So that dip there indicates that the cell needs to reset in some way. So if the cell isn't given sufficient time to reset, that next action potential isn't going to peak quite as high. So because the cell needs time to reset its membrane potential, if the action potentials are too close together, the maximum membrane potential would likely decrease. Part B. Researcher proposes two models, A and B, for using acetylcholinesterase, an enzyme that degrades acetylcholine, to prevent the effect of the neurotoxin. In Model A, that enzyme is added to the synapse. In Model B, it's added to the cytoplasm of the postsynaptic cell. We need to predict the effectiveness of each proposed model. Provide reasoning to support your predictions. So a thing to watch out for here. This question is not asking which one will be more effective. It is asking us to predict the effectiveness of both of them. So we need to address them both. So for Model A, let me rephrase that. Model A will be effective in preventing the effect because receptors on the surface of the postsynaptic neuron are responsible for carrying that signal. Degrading acetylcholine in the synapse will block the signal. For Model B, Model B will not be effective because acetylcholine binds to the outside of the cell. It is going to not have an effect if we degrade acetylcholine inside the postsynaptic neuron. And there we have it. If you folks want to hop into the Google Meet to drop any questions that you have, talk about anything that you are worried about, wondering about, let me know. I would be happy to assist there. Just to protect privacy and all of that, I'd like you not to jump into the YouTube live chat just so that we have all of our bases covered there. While we go on here, we're going to look at one more thing, and I'm going to get this pulled up for us, so give me just a sec. So this one is also from the 2019 AP exam, so it's the most recent exam that was given. And let's go ahead and paste that. And let's zoom this out just a little bit so we can see everything. So this one gives us a lot of data up front, and again, part of dealing with the AP exam is separating out the important information from the extraneous information. So I recommend, before you even start looking at the table, start reading what the question is actually about. So in this one, we're looking at amino acid synthesis in yeast cells. And we've got multiple genes. Researcher conducted an experiment to determine the ability of yeast to grow on media that differ in amino acid content. Researcher tested 2 different haploid yeast strains, each of which has a single recessive mutation, and a haploid wild-type strain. So haploid means one set of chromosomes. So we've got 2 mutants that have a recessive gene, 1 single recessive mutation, and then we've got the wild type that has all of the dominant genes there. So the first part, identify the role of treatment 1 in the experiment. So treatment 1 has all of the amino acids present. So these are all of the amino acids that are needed. So this should serve as a control. All of the yeast, regardless of their deficiencies in synthesizing amino acids, should grow on this. So treatment 1 serves as a control. All of the yeast strains should be able to grow under these conditions. And one of the important things that a control does for us, first, it gives us a pretty quick indication if something has gone awry, because if we don't get growth, we're expecting growth for everything, then we know that something has gone wrong and we can correct the experiment. The other thing that it does is it gives us a baseline for comparison. So because we know what to expect here, this gives us something that we can compare against. All right, we need to provide reasoning to explain how mutant 1 can grow on the treatment 1 medium, but not on the treatment 3 medium. So on the treatment 3 medium, we have all amino acids present except methionine. So the fact that our mutant strain cannot grow there indicates that that mutant strain cannot produce methionine. So because the yeast can grow in the presence of methionine, but not in its absence, this suggests that its mutation hinders its ability to synthesize methionine. All right, part C, yeast mate by fusing 2 haploid cells to make a diploid cell. In the second experiment, the researcher mates mutant 1 and mutant 2 haploid strains to produce diploid cells. Using the table provided, predict whether the diploid cells will grow on each of the 4 media. So in this one, we are getting the genes from both mutants. So what we know about these, mutant 1 can't produce methionine, mutant 2 can't produce leucine. So let's go ahead and I am going to see if I can make this work here. Okay, fine. All right, so we have the diploid strain, So we would expect that the diploid strain can synthesize methionine because it's got a good copy from mutant 2, and that it can synthesize leucine because it's got a good copy from mutant 1. The fact that it's got a recessive copy of each of those from one of the mutants isn't going to hinder its ability to make that. So the diploid one should have pluses for all of those. And let's see here. Yes, of course, this is the time to get particular about the formatting. That is a great use of your time, Mr. Turner. Okay, we're doing this. Alright, and there we go. So the diploid cells are going to have, they should be growing on all of those plates. Alright. Now, we're going to hit the last problem from this one, and I'll have a couple of last minute sort of review reminder pep talk, and we'll wrap this up. So for our last one here, we have changes in morning glory petals during flower opening, and again, you want to look at the question before you really jump into the diagram here. So the petal color of this flower changes from red to blue, and the petal cells swell during flower opening. The pigment heavenly blue anthocyanin is found in the vacuole of petal cells. Petal color is determined by the pH of the vacuole. The model is shown in the table above. So we need to identify the cellular component in the model that is responsible for the increase in pH of the vacuole during flower opening, and describe the component's role in changing the pH of the vacuole. So, looking here, the open flower, we have the differences between these two. This vacuole contains a bunch of hydrogen ions here in the bud, and I notice that in the open flower we have a bunch of potassium ions in there and hydrogen in the cytoplasm. So what I see here also, the difference is the potassium hydrogen transport protein in the bud is inactive, and it is active in the open flower. So that's going to suggest to us that the potassium hydrogen transport protein is going to be involved in this. So the potassium hydrogen transport protein is responsible for the increase in pH in the vacuole. It transports hydrogen ions out of the vacuole and into the cytoplasm. And a quick chemistry refresher, more hydrogen ions means a lower pH, so fewer hydrogen ions means a higher pH. So because more hydrogen ions mean a lower pH, moving hydrogen ions out of the vacuole increases the pH. And then we've got the question. A researcher claims that the activation of the potassium hydrogen transport protein causes the vacuole to swell with water. Provide reasoning to support the researcher's claim. So this one goes back to osmosis and diffusion. So with this one, we have an increase in the concentration of potassium, which is a solute. And when the solute concentration increases, that decreases the water potential, and that will lead to water flowing into the cell and into the vacuole. So that is what's going on there. So let's see if I can sum that up and type that. So the concentration of potassium is increasing in the vacuole. This increase of solute concentration lowers the water potential, causing water to enter the vacuole. All right, there we have it. That gets us to the end of that part, and that gets us a pretty good preview of the AP exam. So just a sec here, and I'm going to check on a couple of things. All right. So I'm going to leave folks with just a couple of reminders, things to think about before the exam. So I know the exam is coming up very, very soon. Before the exam, if you haven't taken other AP exams yet this year, under these new circumstances, go ahead and go through the tech demo, make sure everything works, make sure that the way that you're going to submit things does what you expect it to do. Take a moment to make sure that you've gathered all of the papers and things that you want to have on hand, all of your notes, all of the resources that you want to use there. And beyond that, good luck, everybody. Relax as you go into this as much as possible. I know that the circumstances aren't ideal, but I hope that you are able to do the best that you can, show what you know, and try to, I know that saying enjoy the process of taking an exam is a little bit disingenuous, but I hope that it is as pleasant as possible. All right, that brings us to the end of the session. Thank you, everybody, and I'll see you on the other side of the AP.